I'm going to introduce you, though, to something that, from what we discussed yesterday's meeting, may be quite brand new. Now, the Gulf War population, does everybody know about Gulf War illness? Yes. Okie doke. They had a unique exposure of being in the military service, 1990, 1991, Persian Gulf War. There are some who developed acute illness on the morning of May 11th, 1991. This was the day after the destruction of the Camisea munition bunkers. Sarin gas is now known to be released. This may be an indication of mass organophosphate poisoning with exposure to over 190,000 troops. There's been a chronic onset of symptoms over the past 20 years with progression to disability. 25 to 30% attack rate. I only wish that Congress had a 25 to 30% attack rate, because then we'd get some progress in this. So one of the interesting things here is we've got our original chronic multi-symptom illness definition here based on fatigue, mood, cognition, aches and pains, etc. And then over the years, there have been now about 10 different definitions. The point of this slide is that the original CMI was thought to be too wishy-washy, not specific enough, so they did the Kansas one and added the requirement that you actually have been in the Gulf. There's a whole bunch. Well, I say that because 15% of the veterans who were not deployed but who were in training in the U.S. have come down with these same symptoms. They are not eligible for a chronic fatigue syndrome diagnosis or a Gulf War illness diagnosis because they didn't go to the Gulf. They didn't have the exposure. They don't have anything. Is that fair? Okay, you tell Congress. Okay, let's get back on track here. We've got time to go. Fibromyalgia, they changed their criteria away from requiring tenderness by physical examination to what, to me, sounds like chronic fatigue syndrome, unless you talk to some rheumatologists. Our chronic fatigue syndrome, this is the original Fukuda criteria, which I've used in diagnosing these people, but also when the Canadian criteria came out, we began using them. The common features: fatigue, memory, concentration, pain, tenderness, exertional exhaustion, which is critical, is found in all of these groups. And of course, we all know that this is depression. Because they don't like it, so they're anhedonic, right? It's my job to say it's not depression. And I'm sure you're all against me now. So with these common problems, how can we look at them? Well, they're symptoms, they're perceptions, so it seems to me you got to look in the brain because that's where you create your perceptions. That may not be where symptoms start, that may not be the be-all and end-all, it may not be the original site of etiology, but it's the location where you feel your fatigue, where you have attention networks, I'll show you where those are in your brain, where you have your working memory, which allows you to remember why you walked in here in the morning. Your anxiety, like I had coming up here after that fabulous talk and knowing who's coming up next. I mean, I'm anxious. But no pain or tenderness for another reason that we'll get to. And the exertional exhaustion that is so typical and is the reason we started doing the exercise studies. Now, has everybody here had an MRI of their brain? Okay, so what kind did you have? Did you have the structural scan that looks for the thickness of the cortex or the white matter lucencies? Did you have a diffusion tensor imaging that looked at the integrity and function of the white matter? That's generally damaged in multiple sclerosis, for example. Did you have the blood oxygenation level dependent or BOLD study that shows the region of your brain that is activated when you plan a task or think of something? Was this done at rest or during a task? Did they look at the connectivity, this is what I'm going to be talking about for the rest of the day. Did they look at the bold, the blood flow, plus the white matter together to see if areas are connected and talking to each other? There's other methods here. And finally, the concentration of brain chemicals can be measured by molecular spectroscopy. It's really amazing what these new tools will do. The software is incredible, as it takes a lot of work. I won't pull your line. Okay, so voxel-based morphometry is a fancy name for saying that they're looking for the architecture of the cortex and the white matter. There have been studies in the past in chronic fatigue syndrome showing thinning in the hippocampus, which is a major memory area in CFS. But the problem is you don't know how long these changes in thickness will stay. Particularly with cognitive behavioral therapy, there's one study showing thickening, but you don't know what that really means unless you go in and do a brain biopsy. Not going to do that. At least not at my university. Don't know about yours. Okay, this is, I think, a seminal and very important work that Dr. Qualitative reported 2 years ago by video, and it's Barndens and et al. This is the white matter that is found in the brain, and the white matter forms a skeleton for the brain. So the brain here doesn't look normal to you, but this is all of the white matter that forms the structure. Here's the brain stem down here. And this compares chronic fatigue syndrome subjects with controls. And it's looking for the volume of the different colored areas here. In particular, you can see these red and yellow areas are all significantly decreased in size in CFS compared to controls. This was, I think, very stunning to me to see. And what was even more amazing is they were able to look at the decrease in volume of this white matter in these regions over the duration of the fatigue. And you can see they have a lovely correlation here. The longer the patients had the fatigue, the more their white matter was shrinking. This is not a lesion that you would find in multiple sclerosis, but the location here is very, very critical. We've done a similar study and have been able to reproduce their work. This is in Gulf War illness patients. They showed decreases in the brain stem up in this region here, and the areas that I've highlighted here in yellow, green, and blue, and down here in red, all show decreases in white matter. Now, one of the important areas that you can see here is in the cerebellum. The cerebellum is important because it fine-tunes just about every action you have. Through this area, all of the output of the cerebellum moves through this area. It's called the superior cerebellar peduncle, and it moves sort of from this blue area in through the square, and you can see it very nicely here with the blue area as well. And this is very close to where Qualitativ's group found problems as well. And in talking with Dr. Vincent, this is important because these nerve fibers carry GABA, the inhibitory neurotransmitter, and it just makes me wonder if there's something wrong with these neurons. Okay. Switching gears, the next fancy tool that you can use is BOLD, which tells you about the blood flow in a 1 by 1 by 1 millimeter cube in your brain, or in some cases 3 millimeter cube. Doesn't really matter. Oops. What it actually does is it measures the amount of deoxygenated hemoglobin that's in the blood vessels. I was going to go over all of this, but let me cut to the chase, which is that when you have a signal, it's a 3 to 4% change in what you can measure. This means it's very computationally intense and very sensitive, very difficult to do these measurements. And often you have to average a large number of people together to get a result. The technology is actually getting better. It's suggested that in a year or two we'll be able to get the results from one person, which would be fantastic. Now, how do you regulate this blood flow, which the blood flow indicates what neurons are being activated, what regions of your brain are activated. The neuron acts, and most of them are excitatory and release glutamate. When they release glutamate, the glutamate activates NMDA receptors on astrocytes, a support cell. These cells, the astrocytes, extend from the neurons to the blood vessels, the arterioles. At rest, the astrocytes will normally be producing this arachidonic acid metabolite that's a vasoconstrictor. Closes down the vessels so that you just have a normal level of blood flow. However, when the glutamate is released, the astrocytes release this D-serine, which then stimulates the endothelium to release nitric oxide, which, like other vessels in your body, relaxes the arterioles. The arterioles dilate, and now you have a huge increase in blood flow to a small region, perhaps a millimeter across or less. And that blood flow change is what's measured by the bold signal. So the neuron to astrocyte to endothelium to smooth muscle is the sequence chemically that if there are changes globally in blood flow in the brain, we may be able to modulate it in this fashion. This is an earlier 2006 study by DeLange that looks at a very important region of the brain. This is in chronic fatigue syndrome. It's the dorsal anterior cingulate cortex, or DACC. If you look here, this is a band of white matter, and above it there's a band of cortex, and you can see that in general this area is different when CFS people do a task compared to controls. The effect here is that they are making more errors. When you do intensive statistics, you find that there's a small region here in what's called your executive control network that helps you decide if you've made an error or not and to make changes in your task performance. This area, by lighting up, shows that the CFS people in this experiment were unable to correct their errors. So they can't pay attention. They can't concentrate because this area is not working properly. Big, important issue here, or topic. That was bold. That was just the absolute blood flow. What I'm going to talk about for most of the rest of the time is what areas of the brain are linked together and how are they changed in CFS and other illnesses. And there's a great gap between what I'm going to say and what you've heard before. So we're in London, mind the gap here. This is an observation that came from a student who was at Medical College of Wisconsin. He noticed that when people were resting in the scanner, parts of their brain were actually communicating. And at the time, everybody thought that your brain should be at rest and doing nothing. But in fact, it was talking. Different parts were talking and communicating. That led to this idea that there was resting networks, or networks that were communicating At the time, they didn't understand what was going on. In 2006, though, there was the realization that when you are doing a task, certain parts of your brain are working, but when you stop, a different part of your brain starts to work. And that different part of your brain is called the default network. It's in the front here in the middle and in the back in the middle. I'll show you that in a second. And these areas are switching on and on. Because they're activated at the same time or synchronously, this has been termed functional connectivity between the different regions of the brain. And here's just 4 examples of these networks. Here you can see the brain with 4 different networks, different colors. Back here in the occiput, you have the visual network. This is connections within the early to late processing portions of the occiput when you see something and then start to process the image. Oops. You have your sensory motor areas here, sort of your motor muscle, if you will. The basal ganglia that are very important controlling structures that we'll get back to. And then this very striking default mode in the front and then in the parietal lobe in the center. That, again, this is the mode that when you daydream, mind wander, your mind goes blank, this one switches on. Brainmap is a consortium out of San Antonio, Texas, and they've taken 1/3 of the imaging literature, put it into their computers. They've run analysis of like 10,000 people's fMRI scans, and they've started to look at the areas that are linked within the brain. So, for example, up here, they found a network that involves the amygdala. The amygdala is where your emotions come from, your affect, whether you're happy or sad. We have, let's see, back here will be visual cortex. Here will be the posterior part of that default network. If you like, I can go through all of these. The important issue here is that different parts of your brain communicate continuously. And starting from that first 20, now there's a published set of 70. Would you like me to go through these? Or the next slide, which is the 300 that are coming out shortly? Don't think so. The critical finding is that different patterns of these resting state networks may be indicative of specific diseases. I'll get to that a little bit later. The possibility on seeing this is, does it apply to our Gulf War people? And now that we've started our similar, identical study in chronic fatigue syndrome, does it apply to chronic fatigue syndrome? How do we make sense out of these networks? Well, this is an example of 32 of the networks painted different colors. And I'm not even going to start to describe them here. Just recognize that there are different communications at rest. They then took 18 studies where people were doing specific tasks, like looking at an image and then pushing a button. And that defined areas of the brain that were linked together. Then they added the larger cytoarchitectural or immunohistochemical similar regions, and they came up with what they called 2 intertwined rings. And the key is the red and the red. This is the parietal lobe, which is back here, and the frontal lobe. And this is like the thinking part of your brain. And the blue part here is your sight, your hearing, your touch, and your motor areas. And this visual auditory VSA ring forms a continuous ring. The parietal temporal frontal ring is discontinuous. If we look at it in this way, where I've added the ring, you can see that these regions are all closely connected by short axon connections. In contrast, over here, the inferior parietal lobe that's important for attention, that integrates much of this sensory information, and the frontal lobe that is making decisions and planning are connected by deeper white matter. Here you can see this inferior parietal lobe that acts like a donut hole, and you can see the donut here that will be this VSA ring. By cutting the brain apart like this, you can see the long white matter tracts of the brain that are connecting the frontal to the parietal to the temporal region, forming a loop. You can see they're very long. This is important because we've reported last year white matter abnormalities in our Gulf War people. We hope to determine if those occur also in chronic fatigue syndrome. And if that is confirmed, it will tell us that these long tracts are not communicating properly. So maybe we can fix that. Any questions about that? Okay, good. So is this an easy task or a difficult task? Do you use the same parts of your brain for one task or another? That was the question that was asked in this study. If you have low cognitive load or an easy task, do you use the same networks as a high cognitive load or a difficult task? This will help to explain how some of these networks are, what they're responsible for. Here is the brain scan, the bold and functional connectivity for a low cognitive task, something very simple. You see a letter, you push a button. When you do that, you light up this frontal area on the right that communicates through this white matter to this parietal area back here. This allows you to focus on this task and then do it. You can see what you need to do. You send a message up here. This sends a message back to tell you what you're doing. The insula here, that's just in your temples, has two distinct functions, probably others. In this case, what it's doing is it's checking for errors. Showed you the first study earlier, CFS people don't check for errors. That may indicate a dysfunction in this region. Then finally, at this low cognitive load, people have time for mind wandering, and they are actually able to enter this default mode here where they just zone out. Is anybody zoning out, by the way? Anybody moving into their default modes? It's okay. It'll all be on tape later. Okay. So what happens if you do a more difficult task? A, B, C, D, E. What happens if you do a more difficult task? What letter did I just say? What you did is more difficult. That's called working memory. What you did is you heard me say something, and down in your temporal lobe under here you heard it. You stored it in this region, and then when you were asked to recall it, this part, your executive control network, said, remember, what was just said? It sends a message here to the frontal region, back to the parietal region. The parietal region then tells you what to say that you heard. So you remember ABC, and you send a message to this other circle, that other dual network, to say ABC. This is how your brain works, how one functionally connected network will speak to the other. Here you can see what's called the dorsal attention network. This is critical. If you're looking at something, you have to use these two frontal eye fields. You have to focus using your supplementary motor area, and we go back here to this critical inferior parietal region that is the donut hole. Down here, because it's a more difficult task, you're using your cerebellum and the thalamus and your limbic system, sort of a lower level system that is very important in complex tasks. And finally, before you just had the right frontal parietal system activated, but here you have both sides with a more difficult task. So from this, it's obvious that when you do a low cognitive load task, you're using certain areas of your brain, and then when you have to work harder, you're using a different set of areas. What happens if you can't switch from one to the other? What if you're doing this, but you need to be doing this? What if you can't switch? Then you go into your default mode. You just disconnect. And to do your task, you have to start all over again. And you have to start recruiting new areas here to make it work. Sound familiar? Yes. Good. Okay. Thank you. Thank you. Thank you very much. Okay. Anybody get exertional exhaustion? And do you think clearly when you have exertional exhaustion? Why? Well, let's take a task like the NBAC task. That's like the one I just gave you, A, B, C, D, where you had to remember what I said a little bit ago. That's not exactly an NBAC task, but that's the type of memory, working memory, you have to use. What cognitive networks are responsible for this? This is a very nice little study in healthy control subjects. These people came in, had a resting scan, then they did their cognitive test, then they took the day off, had a resting scan, and then did a second cognitive test. That's all they did on day one. The next day, that was almost identical. They did the resting scan, they did their NBAC test, and then they had 4 hours of intensive helicopter simulation training. They're in a helicopter simulator trying not to crash. Is this a low or high cognitive? I mean, I'd crash the damn thing. I mean, I don't know about you guys. And then afterwards, you do the NBAC test again. So what were the results in these healthy controls, most of them 20-year-olds? Well, for reasons I don't understand, the free day is on the right, and they had no exhaustion. During the day, they had no exhaustion, but by the time they finished their training, they were exhausted. And these are, like I said, 20-year-olds. So if 20-year-olds exhausted, I guess that must be pretty bad. Must be as bad as texting all day. They also had more effort by the end of the day in doing their NBAC test. But what was interesting is they had no decrease in accuracy. They did just as well, but there is a reason why. Now, out of all of the 70 regions I showed you earlier, these are the 7 that were activated significantly and were different between the different baseline resting scans. The anterior default network, the posterior default network here, the visual network, the sensory motor for feeling and moving your fingers, and then your frontoparietal on the right and left sides, and your auditory network. And it's not actually hearing things, but using this for representation, as in recognizing letters and language that's being used here. What they found were only 4 regions in these healthy controls were changed. the anterior default mode was significantly increased when they were exhausted, which meant they were tired, so they were drifting off into this mind-wandering stage while they were doing the task, or while they were just resting. They also were using their early visual network, which means even though they had finished using playing with the helicopter, they were still pretending that they were seeing the helicopter, and they were rehearsing using this part of their brain. But very oddly, they were not using the control networks, either on the right side here, right frontal parietal, or the left frontal parietal regions. So they weren't able to concentrate on the task quite as well as we would have expected at the start. So their conclusion is, was there a functional or a change in their scores? No, there was no change in their scores. But they changed the utilization of their networks after they were exhausted. So that's healthy controls. Let's take a look at what happens in Gulf War people. Last year, Rakib Rehan was here, and I'm happy to report he got into an MD PhD program, and unfortunately will be leaving in about a month. So I'm crushed. Anybody who wants to come work for free, come on down. He did the work of 3 people, so I need 3 people to come on down. Okay, he defined that we had a group of our Gulf War subjects, 1/3. They did the stress test, and then they developed postural tachycardia, which meant they stood up and their heart rates went up. They had brainstem atrophy, and they used the middle part of their cerebellum to try to help compensate to do their NBAC test. In contrast, the other 2/3 had phantom perceptions. That is, they were using the anterior part of their insula, where your conscious sensations enter your perception, where your perceptions enter your consciousness, and they had consistent basal ganglia activation that I'll show you. So these are objectively defined subsets. And I'm damn mad because the people who read my grants last year didn't understand what an objectively defined phenotype was, but next year it will be different. Here you can see something interesting. This is the limbic system in the control subjects before exercise and after exercise. You can see that this area is lighting up in all of these subjects groups, the stop, the start, reversible tachycardia group. You'll also notice that the frontal parietal region is lighting up here in blue. What this means, though, what the difference in color means is that the red network is lighting up, and then at a different time, the blue network is lighting up. So they're trading off. And it's happening in the controls, so that's good. But you look at our start people, and they don't have their frontal parietal control system working. As we have gone along, we've found that this anterior default region was only activated in the stop group. It was found in the controls, but we didn't find it. And the insula was used here in our start group before exercise, and then after exercise, they lost it. We have a technical term for that. It's called going to hell in a handbasket. And you can see here that with their activation of their control networks, they do that again. Now, how do we try to make sense of this? These are, what I'm showing are qualitative responses. The statistical analysis is, to say the least, very intensive and still ongoing. But what we've found is activation of the amygdala and the other parts of the limbic system, and strangely, this anti-correlated control of the action while doing this working memory task. The control people were the only group to use their frontal parietal control regions effectively. The stop people were using their default network, saying that they were becoming disconnected from the task. And they also used their executive control and, for extra compensation, their basal ganglia region. After the exercise, after they developed the exertional exhaustion, the key changes were that now the start people were starting to use their insula. They were recruiting additional areas. And this area, the right parietal, superior parietal up here, which you're not supposed to recruit. So they were pulling out the stops to try to do their task. It was the stop group here pretty much had the same areas of activation. So our interpretation is that our start people show autonomic dysfunction after exercise that's induced by exercise. They have disordered utilization of their cognitive reserves before exercise, and then they recruit extensive areas to try to complete the task. The phantom perception people are using their insula to feel the pain that they feel in their bodies. They use a different part of their brain, the basal ganglia, to compensate and try to complete the working memory task. And they have extensive anti-correlations between their attention networks, their working memory, their executive control networks with this default mode that indicates they're switching in and out and can't maintain their focus. Next is basically complete the statistics, which will take us a while. So why are we doing this? It's because this is a psychiatric disease. Right? Depression? Professor? Yes?
I don't know, you're trying to prove it. It's
okay.
Yeah.
So here we go. This is what the psychiatrists are writing now about different psychiatric illnesses. For depression here, they have some Venn diagrams about the different regions in the brain, these ones that are identified by the functional connectivity. The circle on the left includes the regions that they believe are having dysfunction that is leading to, for example, depression. I won't go over all of the abbreviations here, but you can see for depression it's quite extensive. On the right is the circle that they believe represents the areas that they are effectively treating with cognitive behavioral therapy. And you'll notice that there are no areas left to treat in depression. So I guess we're done. Right? We've cured depression.
Comes and goes.
Yeah, there you go. Especially things like schizophrenia. Only this inferior parietal lobule is left that can't be treated with cognitive behavioral therapy. All of these other areas are effectively treated by psychotherapy in schizophrenia. Okay. This is a better approach to depression. These people, this is a Chinese group, examined 550 regions of the brain in two populations of about 200 people with depression. And they looked at what parts of the brain are linked together, are functionally connected. The size of the circles here represents the strength or number of times that an area is connected. The color represents the weight or correlation coefficient. And one of the keys is that here you have the amygdala as being a key region. And there's also these three red balls. And you can see it's actually 4 when you see it from the top. Over here, I won't go over this except to say that these are different regions of the brain The purple lines are the ones to pay attention to because they connect to your affect, the affective components. In looking at this a little closer, it's not a surprise that you would have changes in your affect and the parts of your brain linked to the amygdala. Here on the left panel, you can see the top 100 regions that are linked in depression. Again, the amygdala is critical here, left and right. But what was new information is here in this panel, these two views, where there were 3 cerebellar regions. Again, we keep going back to the cerebellum here, which we showed to be defective in its connections. And it has defects in communicating to the amygdala, to the frontal part of the brain for executive action and others. The reason this is also of interest is the long white matter tracts that come from the cerebellum that carry GABA, and could this set of nerves be particularly targeted in any sort of illness? Does anybody get anxious? Oh, you're depressed, right? Well, this is a bunch of normal people who took an anxiety test, and they were divided into people who had low anxiety scores and high anxiety scores. The connections between the amygdala down here with these parts of your default network were examined. And what was interesting is the people with low anxiety did not link up or had a negative correlation here, which meant they were shutting this area off. But instead, they were lighting this area up. And this area has extensive connections to your autonomic nervous system. I'm sorry. This area has links that turn off your autonomic nervous system. Here, you can see that you have no connection, and here you have negative links. This area is actually turned off. So this is an indication of changes in the resting functional state during anxiety. It's in your head. Really is. Anybody have gastroesophageal reflux? This similar finding is also found in irritable bowel syndrome. Here's your posterior posterior default network, the precuneus here, and it is preferentially activated in the people who have GERD, gastroesophageal reflux. This is probably because the people are internally focusing on their interoceptive system, the afferent pain coming from their stomach and esophagus. And as a last indicator of the potential for this method, some people feel more pain than others, right? Why is that? Well, in part, here's this area that lit up during the in the anxiety people that I just showed you. This top slide shows that the white matter connects this region to the periaqueductal gray matter here. This is the region that generates the norepinephrine that goes down the spinal cord to where each of your sensory nerves comes into the spinal cord. This works to shut off pain. It's called your descending antinociceptive network, and in people who feel pain more easily, this area has atrophied. And here you can see the diffusion tensor imaging showing the connection between this medial prefrontal cortex and the periaqueductal gray. This an increase in activation and increase in size of this periaqueductal gray is found in asthma. Those people cannot feel short of breath. They don't feel short of breath because they've shut off the signal. Therefore, they don't go to the emergency room when they need to. Therefore, their asthma gets worse, and they come in when they're on death's doorstep. The opposite, I think, is happening in people who have chronic pain states. This area is atrophying. You cannot turn off the pain, so you feel much more of it. And then finally, one of my patients was the president of the American ADHD Society, and I told her she had chronic fatigue syndrome, and she says, "No, I don't, I've got ADHD." And I went over the criteria, and she says, "Well, yeah, but we all have it too." And this makes me start to wonder how many other illnesses are out there that have similar subjective complaints, and are they also suffering from similar types of problems? In this case, this is the set of networks active in ADHD, here in the default network and here in the ventral attention network that are connected. The ventral attention network is very important because if you're doing a task and something flies by, like a bus comes flying through the sky, you want to pay attention to that, and it's up to this network to notify you that this has happened. So, with that in mind, I think this shows that there's great potential for this methodology in looking at the function of these different networks. I didn't mention it, but the orbital frontal area just above your eyes may be an area where you decide if you want to continue doing something. That's not really the way that it would work, but it's been proposed as being a fatigue center by workers in Osaka. But alterations in the functional connectivity of each of these other networks, I think, is going to turn out to be very critical in understanding the symptomatology of these chronic fatigue syndrome, ME, and these other illnesses. I'd like to thank Richard and Pia. Thank you all, and hope there's no hard questions.
Thank you very much. I have one question for you specifically sent in. Do the apparent lesions found in brain scanning indicate damage? Is this permanent or reversible?
Based on the Barnden study, the first one that I showed of atrophy that correlated with the years of fatigue, I would think this might be potentially irreversible. The problem is that you don't have a lot of neural stem cells that could go into a damaged area and produce new neurons. The trick is to retrain your brain to use the areas that are working, use them more efficiently so that you don't lose any more function, and the parts of the brain that you do have, you're using more effectively.
Okay.